Breeding methods of self-pollinated crops.
Self-pollination is the transfer of pollen from the anther to the stigma of the same flower or another flower on the same plant, resulting in fertilization without the involvement of another plant. Common self-pollinated crops include wheat, rice, pea, barley, and soybean. Breeding in self-pollinated crops is important for developing pure lines with stable and uniform traits. It helps in improving yield, quality, disease resistance, and stress tolerance while maintaining genetic uniformity over generations. Self-pollinated crops are characterized by high homozygosity and a stable genetic makeup, which ensures uniformity across generations. They show less genetic variation and allow pure lines to be easily developed and maintained.
Breeding objectives of self-pollinated crops are homozygosity and superiority. Superiority in traits such as yield, quality, pest resistant, adaptation to environmental conditions.
Mass selection is a simple and traditional plant breeding method in which a large number of superior plants are selected from a population based on their phenotypic performance. The seeds of these selected plants are mixed and used to raise the next generation. This method improves the average performance of the population and is mainly effective for traits with high heritability.
Pure line selection. It involves selecting the best individual plants from a self-pollinated population, testing their progenies for several generations, and finally releasing uniform, stable, pure lines. Advantages: produces highly uniform and stable lines. Disadvantages: time-consuming and limited scope for further genetic improvement.
Pedigree method. It involves hybridization followed by selection of superior segregants in each generation, while maintaining pedigree records, leading to evaluation and release of improved varieties. Advantages: effective for combining desirable traits. Disadvantages: time-consuming and requires detailed record keeping. Bulk method. In the bulk method, generations from a cross-population are advanced in bulk with little or no selection until later generations, usually F4 or F5, for natural selection. Pros: simple, low-cost, natural selection operates. Cons: slow progress and less effective for low heritability traits.
Single seed descent method. Advancement of generations using single seeds per plant. Steps: crossing, single seed selection per plant in each generation, selection and evaluation in later generations. Benefits: fast development of homozygous lines. Limitations: no early selection and requires controlled handling of plants.
Back crossbreeding. Repeated crossing of hybrid with one of its parents. Hybridization with recurrent parent. Selection of desired traits. Repeated back crossing. Evaluation and release. It is used to transfer a specific resistance gene from a donor parent into a well-adopted variety by repeated back crossing while retaining the desirable traits of the recurrent parent. Mutation breeding. Inducing genetic changes using mutagens. Mutagens are physical, radiation, chemical, EMS, biological, viruses. Selection of mutagens involves choosing physical or chemical agents that effectively induce beneficial genetic variations in a target organism.
Molecular markers help identify and track desirable genes in plants for precise breeding. Marker-assisted selection, or MAS, accelerates breeding by enabling early selection and reducing reliance on phenotypic evaluation, and it is effectively used to develop plants with improved disease resistance and enhanced quality traits.
Comparison table. Breeding methods for self-pollinated crops. Major breeding methods include selection, hybridization, mutation, and backcrossing to improve crop traits. Integrating conventional and modern techniques enhances efficiency and precision in crop improvement.
Biotechnology and genome editing enable targeted trait improvement and faster development of superior crop varieties. Thanks for the time.